Okay, so good evening, everyone. So today we are in the week 9 tutorial session of the NPTEL course, Genetic Engineering Theory and Application. In the last class, what all topics we were discussing? So in the last class, we learned about the different purification techniques or the different chromatography techniques. So starting from the gel filtration chromatography, when we learned about the ion exchange chromatography, hydrophobic interaction chromatography, and the affinity chromatography. So all these chromatographic techniques were separating our protein on the basis of the properties of a protein. So a protein has several properties. It has a charge. It will carry a charge. It will have a shape. It will have a particular size, and it will also have some. It can also have some affinity. So if it is an enzyme, it will have an affinity for a substrate. If it is an antibody, it will have affinity for an antigen. And also it will have hydrophobicity, okay, depending on the number of hydrophobic amino acid residues which are present. So it will also have will also have a hydrophobicity. So utilizing these different physical properties of a protein, physical and chemical properties of a protein, we can separate protein using different types of chromatographic techniques. So in the last class, we covered all these topics. We quickly discussed these topics which were covered in your lectures. So in today's session, so we will learn about the characterization of the purified product. So once you have purified your protein, then how will you know that the protein which you have purified, that is your protein of interest, or whether you have purified your desired protein of interest, how will you know? Because anything, so all these biomacromolecules, they all appear white. They are transparent. They appear like water. You will not be able to know unless you have some biophysical, carry out some biophysical techniques to characterize these proteins. Whether so we will in this session. So in the we will learn about these different techniques by which we can characterize our product. Okay, so let's start today's session. Okay, so yeah, good evening, everyone. I'm Akash Jyoti Pathak, and I'm one of the PMRFTs for this NPTEL course, Genetic Engineering Theory and Application. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. So today we are in the week 9 problem solving session. So let us first have a quick recap of what all topics were covered in week 9. So in week 9, we are mainly learning about the different methods by which we can characterize our protein, or we can characterize our product. Okay, how we can characterize our product. So this is again divided into two parts. So in this week 9, mainly the first part of this characterization was covered, and in the week 10, we will look out. We will again learn about further characterization techniques like spectroscopy so there are different kinds of spectroscopic techniques by which we can characterize our protein of interest. To understand how pure our protein of interest is, how whether the protein which we have purified is the protein which which is of our interest. So we will learn about these different spectroscopic techniques as well. Okay. How do we characterize? So in week 9, the two main techniques that were covered. One is the gel electrophoresis. So how we can visualize our or So one way of actually visualizing our protein of interest is by running a gel electrophoresis. So this gel electrophoresis, mainly what we do in labs are we run a SDS polyacrylamide gel electrophoresis. We will learn about the principle, and this SDS page separates protein mainly on the basis of their molecular weight. On the basis of their molecular weight. So if we know the protein of interest, then based on the number of residues which are present, it will have a particular molecular weight. Okay, what are the residues and what is their sequence? So depending on it, it will have a molecular weight. So when we run an SDS page, uh, we uh, we load our sample on the gel, and along with it, we also run a protein marker. Depending on the where we get our protein band and the protein band that we get to which marker this is corresponding, we can actually have an estimate that what is the whether we have got the whether we have been able to purify our protein of interest. And also this SDS page will also tell us about the purity of the protein which we have purified, whether it is having a single band. If it is having a single band, then it is a pure protein, while if it is having multiple bands, then it would suggest that the protein has not been purified properly. Okay. uh, and the next technique is we learn is Western blotting. Okay. So we will mainly learn about these two techniques in today's problem solving session. So how we can characterize our product? So based on the molecular weight, we can either, okay. So so we can characterize our product based on the molecular weight. So if we know the protein of our interest, so uh, if we have overexpressed a protein, we will know the molecular weight of the protein. We will know about its we will know about the func- we will have we will know everything about this. So last class we discussed that how we can know about the PI of a protein, what is the hydrophobicity of a protein, what is the number of aromatic acid, amino acid residues in a protein. So if you know your protein, you can directly how will you know the different physical and chemical properties of your protein. So you directly take the faster sequence of the protein and you put it in the XPASI prot param uh, determine. So you go to this XPASI website and you enter your faster sequence, and then you click on the compute parameter. So that will give you, so you can prot param, you can Google it, and when you open it, if you enter your faster sequence, then this expressive prot param will give you all the different properties of your protein. So the number of amino acid residues that are present, what are the number of basic amino acid residues, what is the molecular weight of the protein, what is the PI of the protein, what is the hydrophobicity of the protein, acidic, basic amino. So every information you will get about your protein. So now you know you have an idea that what is the molecular weight of your protein. So so now you want to characterize, you want to understand that the protein which you have purified, the sample you have your purified sample. Now you want to know whether your purified sample has the intended protein of interest. So you can do a gel filtration chromatography. So we learned about the gel filtration chromatography, that it will separate protein on the basis of the size and shape of the protein. What is how will you so just by running a gel filtration chromatography, how will you know whether you have your desired protein of interest? So what is done in gel filtration chromatography is you run different standards, yeah, through your column. What do you mean by standards? So by standards we mean that there are a few standard proteins like lysozyme, then there is RNase A, then there are ovalbumin. So all these different proteins have a particular molecular weight. So you inject each of these proteins one by one into the column, and depending on their molecular weight, they will be eluted at different time. Okay, so for example, lysozyme has a molecular weight of 15 kDa. This has some. You can also run BSA. BSA has 66 kDa. So well, once you run all these different proteins, you will get different peaks. For lysozyme, you will say you are having a peak here. Then so lysozyme has a small molecular weight, so it will be eluted much later. So you will have a peak somewhere here. BSA has 66 kDa, it will be eluted somewhere here. RNAs will be eluted, say, somewhere here. So now so this is the time and this is the absorbance. Okay, so for each of this protein, what you will get? So for each of this protein, you know the molecular weight, and you have calculated the time at which these proteins are getting eluted. So now you can draw a standard curve. Okay, how will you draw a standard curve? So now you have got two information here. You have got the information about the time at which different molecular weights are getting eluted. So if you plot the molecular weight on the x-axis and the time on the y-axis, then say for example, this is 15 kDa, this protein is getting eluted somewhere here, say. Your 66 kDa protein is getting eluted here. Then something is getting eluted. We will get a standard curve like this. Okay. No, so now depending on, so now you inject the protein which you have purified inside the column. It will be eluted at a particular time. So now you can, so now if once you have plotted the standard curve and after you inject your protein of interest, you will again have a peak. Your chromatogram will give you a peak. Now, depending on the time at which your protein is getting eluted, you can know the molecular weight. From this time, from this standard curve, you can get the molecular weight of your protein depending on what time your unknown protein or your protein of interest is getting eluted. You can estimate the molecular weight of the protein. So that is how your gel filtration chromatography will give you an idea that what is the molecular weight of your protein. Okay, usually what is done in that? So whenever people run gel filtration chromatography, and if they want to estimate the molecular weight of the unknown protein, then first of all they inject multiple standard proteins into the column, and so different columns will actually give you different elution time. So you have to plot a standard curve, and then for your unknown protein or your protein of interest, you can depending on the time on which it is getting eluted, you can extrapolate, and you can, from the standard curve, you will get to know what is the molecular weight of your protein of interest. Now, the second method is by running an electrophoresis. So as I was mentioning that electrophoresis, we will learn about them. So you can have different types of electrophoresis methods, the native electrophoresis and the um, denaturing electrophoresis. So a native electrophoresis where you do not have SDS, and your denaturing is where you add SDS and other denaturing agents like DTT, beta mercaptoethanol. Other method is by mass spectrometry. So mass spectrometry will give the absolute molecular weight of your protein. Okay. So mass spectrometry or MALDI, you can do a MALDI, and then you can get your get the molecular weight of the protein. Now, if you want to characterize depending on the functional group of a product, so there you have to go for IR and UV visible spectroscopy. So we will learn about this. So you will be learning in this in week 10 about these different spectroscopic methods. And the third is the sequence of a protein. So just by sequencing, you can also characterize your product. So these are the different methods. So if you have to characterize a product based on the molecular weight, then what all techniques are you going to use? So you can use gel filtration chromatography or SEC, size exclusion chromatography. You can use electrophoresis. You can use mass spectrometry. And then if you have to characterize on the basis of functional group, then you can do it by using IR and UV visible. And if you have to characterize on the basis of sequence of the protein, then you can go for sequencing. So there are different sequencing methods. We learned Edward sequencing and Sanger sequencing, Edmund sequencing, Sanger sequencing, all these different sequencing methods can be used. Okay, so let's quickly discuss what is electrophoresis. So electrophoresis is what? So when you have applied an electric field and you want to when you are running a sample in the presence of an electric field, then that is called electrophoresis. So suppose you have a charge particle which is having a charge Q. So in presence of the electric field, it will experience a force what? So a charge particle when it is applied to an electric field, then it experiences the force which is QE, F equal to QE. So this is the charge of the protein multiplied by the electric field that has been applied to it. So that is the force applied by an electric field on a charged sample. Okay, so that is QE. Now, other than this sample, this protein sample has a particular shape and it will have a particular shape, isn't it? So depending on the shape, it will also experience a frictional force. So the frictional force will be what? It will oppose the, so this is your frictional force and this is the force applied by your electric field. So the electric field um, uh, will try to move it along the direction of the electric field, will apply a force along the direction of the electric field, while this frictional force will oppose the mobility of the charge particle. So it will act in an opposite direction. So what happens is these two forces, this frictional force and this and the force applied by the electric field, that will be balanced because when your charge particle will move through the gel, these two forces will balance each other and the charge particle will migrate uh, depending on these two forces. Okay, so your frictional, what is this frictional force? So frictional force is the viscosity force. Basically, it is 6 pi eta RV. RV. So eta is what? It is the coefficient of viscosity, and R is what? R is the radius of the particle or the radius of the sample, and V is the electrophoretic mobility. Okay, so the friction force is the will oppose the movement of the charged particle, and it is equal to F into V. So F is the friction coefficient, and V is the velocity of the electrophoretic mobility. So the movement of a spherical through a liquid medium of viscosity eta. Eta is your viscosity of the media. So the viscosity of the media is the it's called eta and denoted by eta, and the frictional coefficient F will be given by what? It is 6 by eta RV. Okay. So now you want to if you want to derive this equation. So when your frictional force will be equal to the force applied by the electric field. So then what will happen? This 6 by eta RV will be equal to QE. So from this equation, you can calculate the mobility of the charge particle. So mobility of the charge particle will be the V. V is the velocity with which the charge particle is moving. So that is your electrophoretic mobility. So it will be equal to QE by 6 by eta R. So you can see here that this electrophoretic mobility is depending on what? It is depending on the charge and the mass of the particle. Okay, so R is basically what? R is determining the mass of the particle. Okay, so that is why your electrophoretic mobility is the ratio of the charge of the particle and the mass of the particle. Okay. So new. So this is V or electrophoretic mobility. V is equal to what? It is ZE by 6 pi eta R. So that is why V is related to the charge by radius of the molecule. So for a globular protein, we can consider that the radius of the molecule is related to the molecular mass. Okay. So radius will be dependent on the molecular mass. Okay, so this one was covered in the lecture. So how the radius of gyration or the hydrodynamic radius is dependent on the molecular mass. So that is why you can substitute this for mass. So now, so this will be the relative mobility u dash. Okay. So you can see here that so when you are running any charge sample through the gel. So why we are considering charge sample? Because your protein is having a charge, isn't it? It is having a charge and it is also having a mass. So the charge will depend on what type of amino acid residues are present in the protein. So and also what is the pH in which the protein is present. So that will give a charge to the protein, and the protein will also have a mass. Okay, so whenever you will run a protein sample through, or when you are doing electrophoresis of a protein sample, your protein will, the mobility of your protein will depend on both charge and the mass of your protein. Okay, so so that is one thing you need to understand. Okay, but when you are doing an SDS page, then what happens is your protein is getting separated only on the basis of mass and not on the basis of charge. So why is it so? We will understand when we learn about the principle of this technique. Okay, because if you because since both these factors are controlling the mobility of the protein, you will if you are running multiple samples, then you have to actually eliminate one of these factors so you can you can understand that what is the mass of the protein. Okay, so otherwise when you run an electrophoretic gel, uh, you also run a ladder, protein ladder along with it. So if you do not eliminate one of these factors, so we in an SDS page we eliminate the charge, so that now we can now only the mobility will depend on the mass of the protein. So the mass, depending on where we are getting the protein band, that will be the molecular weight of the protein. Okay. Okay. Now there are two different kinds of electrophoresis. One is the zonal electrophoresis and another is the moving boundary electrophoresis. So these are covered actually. So moving boundary electrophoresis is when we are separating your sample without a without any supporting media. So what is done? So this is actually a U-shaped U-shaped tube, and this is filled with a buffer. Okay, this is filled with a buffer, and then you apply your sample over here. You apply your sample over here. Now, depending on the charge of the sample, whether it is negatively charged or positively charged, it will either. So if your sample has mixture of different analytes, then what will happen? Your positively charged analytes will move towards the cathode and your negatively charged samples will move towards the anode. Okay, so what is happening over here? So the electrophoresis is carried in solution without a supporting media. So there is no supporting. This is actually this U-shaped tube. This is filled with the media, and on the basis of the charge, it is getting separated. It will either move to a positively charged molecules will move towards the cathode and the negatively charged molecules, they will move towards the anode. Okay, so the sample is dissolved in the buffer and the molecules move to their respective counter-charged electrodes. So sample is located in the middle of the U tube. So this is your, you apply it on the Okay, you apply it in the middle of the YouTube, and then depending on the charge, what will happen? Now this apparatus will be connected to the external power supply, and the charge particles will move to the opposite electrode as they pass through the refractometer. Change can be measured. Okay, so your particles, charge particles will get separated, and as a result, one side there will be concentration of positively charged particles, and on the other side there will be concentration of the negatively charged particles. As a result, there will be some change in refractive index that can be measured using a refractometer. Okay. Now, the disadvantage is that the resolution of this technique is very low due to the mixing of the sample as well as overlapping of the sample component. So now these ones, suppose there are three different samples and all of these are positively charged. Then what will happen? Then all these three will be present at the same position, isn't it? So the resolution is or the samples will not be separated. So it is not able to separate these three different components in the sample, the A species, B species, and the C species. So that is why the resolution is less for moving boundary electrophoresis. Okay. Behavior of molecules in an in an electric field. Okay. Now zone electrophoresis is the one where we have this agarose gel electrophoresis and the SDS-PAGE gel electrophoresis. Okay, so zone electrophoresis, the difference is that there we were not using any media, but here we are using a supporting media. So this supporting media can be anything like it can be a absorbent paper, it can be a starch, it can be agar, it can be polyacrylamide. So for SDS-PAGE, it is polyacrylamide. For agarose that we use to analyze our DNA samples, that is agar, or it can be absorbent paper as well. Okay. The major advantage of presence of supporting media is that it minimizes mixing of the sample and immobilization of the molecule after electrophoresis. Okay, so here your sample is getting immobilized, isn't it? So, and it makes the analysis and purification of the molecule from the gel much easier than the moving boundary electrophoresis. So directly we can visualize the gel, and then we can determine the purity, we can analyze the molecular weight, we can use it for other applications, which we will learn in the coming slides. Now there is a horizontal gel electrophoresis. So if you have done an agarose gel electrophoresis, then you will see that your agarose gel electrophoresis is a horizontal gel electrophoresis, because here your DNA is moving from the towards the positive electron. It is moving towards the positive electron. Your DNA is negatively charged, and when you apply the electric field, then it will move towards the positive electron. So that is horizontal gel electric. So if you see the setup, then you will understand it is horizontal. There is a horizontal movement of the molecules, while in vertical gel electrophoresis, your sample is moving in this direction, in the vertical orientation. And this electrophoresis is a discontinuous way. With buffer in the upper and lower tank connected by the gel slab. It has multiple modifications. So why is it called a actually your SDS-PAGE is also called a discontinuous gel electrophoresis system. Why is it called a discontinuous gel electrophoresis system? Because you have two different media, actually supporting media. One is your stacking gel, and then you have the separating gel. That is why it is called a discontinuous gel electrophoretic system. Okay. Now, the frictional coefficient in electrophoresis for a spherical particle moving through a liquid medium is given by what? So just now we learned about this electrophoretic mobility. So the electrophoretic mobility of a particle will depend on what? It will depend on the force which is exerted by the electric field, which is Q into E. And it will also experience a frictional force which will oppose the electrophoretic or which will oppose the mobility of the particle. So your frictional force is given by 6 pi eta RV. So R is the viscosity or the coefficient of viscosity, and eta is eta, and R is the radius of the particle, and V is the velocity with which the particle is moving. Okay. So for a spherical particle, it is 6 pi eta RV. That is this one. Okay. It is actually derived from the Stokes law. If you have learned about the viscosity in class 11, you will know that this Stokes law, that what is this Stokes law says that when this sample is moving um, through a liquid media, then the two forces will balance each other. And the point at which these two forces balance each other, at that point we can calculate the mobility of the particle. Okay. That the friction force experienced by a sphere is directly proportional to the fluid's viscosity and the radius of the sphere, and a constant factor which is 6 pi. The relationship indicates that as the viscosity or radius increases, the frictional force opposing the particle's motion will also increase, impacting its electrophoretic mobility and overall movement through the medium. Okay, so if there is a spherical particle and if there is an extended particle, then which one of these two will experience a more frictional force? The one which is having a more extended shape compared to the one which is having a spherical shape. Okay. So now, what is polyacrylamide gel electrophoresis? So basically polyacrylamide, so we learned about electrophoresis. Okay. So now we will learn about what is polyacrylamide gel electrophoresis. So polyacrylamide is basically the component with by which you have used for making the supporting media. So if you have used polyacrylamide for preparing the supporting media, so just now we learned that the zonal electrophoresis is done using a supporting media. So if your supporting media is polyacrylamide, then we call it polyacrylamide gel electrophoresis. If it is agarose, then we call it agarose gel electrophoresis. So basically polyacrylamide is what? It is a polymer of the acrylamide monomer. Okay, so the polyacrylamide gel electrophoresis of is done usually for proteins. And this polyacrylamide gel that you prepared, that has a particular pore size. Okay, and this the pore size, depending on the pore size, that will determine the resolution of separating the samples. Okay, so you can actually prepare different percentages of polyacrylamide gel in lab. So you can either prepare 12% polyacrylamide gel, you can prepare 15%, 10%. So you can you can prepare any percentage of polyacrylamide gel. So depending on what is the concentration of your polyacrylamide gel, um, that will control your separation, and that will determine the pore size of the polymer. Okay, so the higher the concentration of the polyacrylamide gel, the more the pore size will be more will be lesser. So if it is an 18% gel, then it is then the pore size. So this has the pore size, isn't it? So the pore size will be very less. While if it is a 10% polyacrylamide gel, then it is having very large pores. So your particle will move very fast compared to when it is run on an 18% gel. So basically here also what is happening? So similar to what we had learned in gel filtration chromatography in a sense, because in gel filtration chromatography also the stationary phase was having a particular pore size. So the gel bead had a pore size, and depending on the pore size, the particles are getting separated. So here also the molecular separation is based on the gel filtration, which is the size and shape as well as the electrophoretic mobility. Okay. So, however, electrophoresis differs from gel filtration in that. So, what is the difference between a gel filtration and a electrophoresis? So, in gel filtration, the particles which are larger in size, those were getting eluted earlier compared to those which were having smaller size. And the opposite is true for polyacrylamide gel electrophoresis. So when you do an electrophoresis, the one which are having a smaller size, those will have a higher electrophoretic mobility compared to the ones which are having a larger size. Okay. Now the pH of the gel is high enough so that nearly all the proteins have net negative charge and moves towards the positive electrode when the current is switched on. So the pH of the gel is actually, so we will learn about this, the stacking gel and the resolving gel. So the resolving gel has a pH of about 8.8. So at the pH 8.8, most of your protein will have negative charge. Okay, because your pH is greater than the pI. So that is why your protein will have negative charge. Now molecules of similar size and charge will move as a band through the gel. And following electrophoresis, the separated bands will be visualized in the gel by an appropriate technique, such as soaking the gel in solution or stain the band slightly to the protein. So we will learn about these things. So we will stain it in case of SDS-PAGE. And if we want to do a blotting, then we can again soak the gel and then probe it using our antibodies. Now SDS-PAGE, what is the SDS-PAGE? So SDS-PAGE, what it is doing is, in SDS-PAGE, we are running, so we have the supporting media. So the supporting media is made up of polyacrylamide gel. Okay, so we have made the supporting media which is of polyacrylamide gel. Now what is done? Is that this this gel actually, when you prepare the gel, you have different components. You add different components in the gel. So first of all, you add this polyacrylamide. Okay, you add the polyacrylamide. Then other than this, what is added? Other than it, you add SDS to your gel. And then what you add? You add tris. Then you add APS, and then you add TIMET. So you add different components for preparing your gel. So when you're preparing the gel, you add both polyacrylamide and you add SDS as well. So what is the purpose of adding SDS to your gel? So SDS, if you see the structure of SDS, it is amphiphilic molecule. What is an amphiphilic molecule? So amphiphilic molecules are those which have both hydrophobic and hydrophilic region. So this is it is SDS is what? It is sodium dodecyl sulfate. So it has 12 hydrocarbon chains, or it has a hydrocarbon chains which are made up of 12 carbon units. Okay, so that is why this hydrocarbon chain, this dodecyl hydrocarbon chain is your hydrophobic region. Okay, it is our hydrophobic region. This is basically a detergent. So detergents will have what? It will have a hydrophobic region and it will have a hydrophilic region as well. Okay, so that is why it is amphiphilic. So now this SDS, what is it does? It is actually a chaotropic agent. It is a chaotropic agent, meaning that when you add the SDS to your protein, this SDS will break the hydrophobic, all the non-covalent interactions that are holding the protein together, that will be disrupted. And your protein will get converted from a linear it will lose its native conformation. So when you add SDS to your protein, so for example, your protein was globular in shape. Now when you add SDS, your protein gets linear in shape. Okay, and also SDS gives charge to the protein. It will add uniform negative charge to your protein. Okay, now other than SDS, so this is the function of SDS basically. So SDS, what it is doing, so it is sodium dodecyl sulfate is a technique for separating mixture of protein based only on the size. So why we are adding SDS? So as I was mentioning when we were discussing about electrophoretic mobility, that your electrophoretic mobility is depending on both the charge of the protein and the size of the protein. Now, if we want to measure the size of the protein, then your size is also dependent on the if we do an electrophoresis, then we will never be able to estimate the size of the protein, because the mobility or the band, that the protein band that you observe, that will also be dependent on the charge of the protein. So if it is negatively, if it is say for example, if it carries a 3 negative charge, then irrespective of its size, so not only so even if it is a very if it has a very large size, because of the very high negative charge, it will migrate much faster compared to the one which is having the same size, but its charge is too negative. Okay, so that is why we have to eliminate one of these factors. So when you are adding SDS to your protein, what is your SDS doing? It is actually eliminating the charge factor. Okay, because what it is doing, it is adding uniform negative charge to all the to your protein sample. As a result, your charge to mass ratio actually now it is getting separate on the basis your of your mass to on the basis of mass per unit charge. That is the basis on which your protein is now getting separated. So your separation is only based on the size of the protein and not the charge. Okay. So the main function is that it produces an even charge across the length of the protein that has been linearized. So it is adding and how does it provide a uniform charge? So this is because that SDS interacts with protein and in a ratio of about 1.4 gram SDS per gram protein. So about 1 SDS molecule for every 2 amino acid residues. So for every 2 amino acid residues, there will be one negative charge that will be added to the protein. And because of this large negative charge, the SDS imparts the or it masks the intrinsic charge of the protein. So so every protein has an intrinsic charge depending on the number of acidic and basic amino acids that are present and in which buffer it is present. That will determine the charge of the protein. But when you are adding SDS, all the protein molecules will have a uniform charge. And so the net result is that the SDS treated proteins have similar shapes and charge to mass ratio. So your SDS is serving two purposes. One is it is linearizing the protein. So the shape of the protein is getting one factor is the shape as well, isn't it? So the shape is getting eliminated because all the proteins are now having linear shape, and the second factor that is getting eliminated is the charge. So all the proteins are going to have a uniform charge. So it is eliminating these two factors, and as a result, what is happening when we are doing an SDS page, the protein is getting separated only on the basis of the size of the protein or its mass. So it is having similar charge to mass ratio. Okay, charge to mass ratio, E by M ratio, we can say. So E by M ratio will be same for all the different protein which is present in your sample because E by M ratio is what? Depending on the mass, it will have more charge. So actually, we do not say that the mass is same. We say that the charge to mass ratio is same. Why? Because the one which is having a larger size will have have equivalent number of negative charges. One which is having a smaller size. One which is having a smaller size that will have its equivalent number of negative charges. So as a result, your charge to mass ratio will be same when you add SDS to your protein. Okay. So by first loading them into a gel made of polyacrylamide and then applying an electric field to the gel, the SDS coated proteins are then separated. The electric field acts as the driving force, drawing the SDS coated protein towards the anode, with larger protein moving more slowly than the smaller protein in order to identify protein. So what is done is basically I'll just so when you are doing an SDS page, what is done is first you have to prepare your gel. So when you prepare a gel, so you have two gel components. One is the dissolving gel and another is the stacking gel. Okay. So now the composition of the resolving gel differs from that of the stacking gel. So your resolving gel has a pH of it has a pH 8.8. Why? So what are the components? First of all, let's understand what are the components which are added for preparing the gel. So the components which are added is first of all is your acrylamide. So you can prepare different type of gels. You can either prepare a 12% gel. You can prepare a you can prepare a 15% gel, 18% gel. So any percentage of gel you can prepare depending on what is your purpose. So if you want to separate proteins which are having very similar sizes, then you will obviously use a gel which is having a higher percentage. So if you use a 18% gel, and if you want to separate samples which are having sizes, say example, 11 kDa, 11.5 kDa, 13 kDa, 14 kDa. So if you want to separate these four different proteins on a gel, then you have to run a very high percentage of gel. Okay. So you will add acrylamide depending on what type of gel you want to prepare. Either you can prepare 18% or you can prepare a 12% gel. Then next, what you will add? Next, water is added. Then your buffer, your trace buffer is added. So for preparing resolving gel, the trace buffer, which is having pH 8.8, is added. Okay. Now, in the next step, what is added? One is APS, and that is the last thing that is added is timid. Okay. And for preparing the stacking gel, so your stacking gel has a fixed acrylamide concentration or polyacrylamide concentration. Polyacrylamide, it has 5% polyacrylamide. And other than this, you add water and all these other components. This tris, but the tris you use for preparing a stacking gel has pH 6.8. Okay, pH 6.8, and then you add APS and TMED. Okay, so basically the polyacrylamide that you have in the lab, that is actually a that is actually a mixture of bisacrylamide and acrylamide. Okay, bisacrylamide and acrylamide. So when you prepare so what is usually done in lab is you prepare a 30% stock solution, 30% polyacrylamide stock solution. So when you prepare this 30% polyacrylamide stock solution, so you add 29 gram of polyacrylamide and 1 gram of bisacrylamide. So that is the composition that is used for preparing a 30%. So for preparing 100 ml of 30% polyacrylamide, what will you add? You will add 29 gram of acrylamide and 1 gram of bisacrylamide. So the function of bisacrylamide is that it is a cross-linking agent. So your acrylamide monomers will be formed, and these acrylamide sheets of acrylamide monomers will be attached by this base acrylamide. So there is this CH2 group that will attach these. So it is cross-linking agent. So remember that this bisacrylamide is a cross-linking agent. It will cross-link the polyacrylamide units. Okay. So and it will form a mesh-like structure. Okay. So why pH 8.8 and why pH 6.8 we will understand in the upcoming slide. But just remember that for preparing the gel, you need two different you have two different gel. One is the resolving gel and another is the stacking gel. And again, what you do is when you are loading your sample, you mix your protein sample with a loading dye. Okay, you mix your sample with a loading dye. Now this loading dye has also has SDS. It has beta mercaptoethanol. It has and it has glycerol. Okay, and it also has this it has bromophenol blue. Okay. So when you are loading the sample on the gel, so what you do is you mix your sample with the loading dye. You have loading dye, which is there in your lab, and you mix the sample with the loading dye. Now this loading dye, the loading dye that is prepared, that has these different components. It has SDS, it has beta mercaptoethanol, it has glycerol, it has bromophenol blue. Okay, so we will understand that what is the function of each of these components. So, your loading dye also has SDS, and the gel that you prepare that also has SDS. So your SDS is playing a vital role. Why? Because it is linearizing the protein first, and second, it is providing a uniform charge, or it is providing a uniform charge to all your protein samples. So all the other different proteins that are present in your sample, they will have a uniform charge to mass ratio. Okay. So this is how the gel, SDS-PAGE gel looks like. So these are your markers. So this figure is taken from the book Void and Void. So this one lane and two and the second lane. And the first lane and second lane, you have run the protein ladder. So protein ladder is commercially available when you load on when you load it on the gel and run it, then it will give you different bands. And the bands correspond to each of these bands correspond to a particular molecular weight. So now when you run your protein of interest, then by comparing it with the protein ladder, you can estimate that what is the molecular weight of your protein. So for example, this is the band. So this band corresponds to around 66 kilodalton. Okay. Now, if you consider this band is somewhere between 31 kDa and 45 kDa. So by running an SDS page, you can analyze what is the molecular weight of your protein, how pure is your protein, all this information you will get from this SDS page. Now, so this is 10, 12 is again a ladder and 17 is also a ladder. Now, how do you get the molecular weight of a protein? So absolute molecular weight, how will you get? By plotting this molecular mass versus the relative mobility. So this thing we will discuss in the upcoming slide. So no need to get confused. Uh, so let's go one by one. Okay. Now, what are the components of an SDS page? So the first component is SDS. SDS is what? SDS is a detergent, basically. It is sodium dodecyl sulfate. So it has 12 carbon uh, hydrocarbon chain. So there are 12 carbon units which forms this long hydrocarbon chain. And then there is a hydrophilic group which is formed by the sulfate. So it is composed of a hydrophilic group with a net negative charge and a long hydrophobic chain with neutral charge. So the hydrophobic chain, this is your hydrophobic chain and this is your hydrophilic region. So it is an amphiphilic molecule. Okay. And it will provide a negative charge to your protein. So the more concentrated the gel is. So as I was mentioning that you can prepare different percentages of gel, 18%, 15%, 12%. So the more the concentrated your gel is, the slower the protein will traverse. Okay. So if you are running an 18% gel, then you have to run it for, say for 3.5 hours or 4 hours, you have to run for resolving your protein. Okay. But if you are running if you are running a 10% gel, then that your gel will be running 1 hour or less than 1 hour. Okay. So the loading buffer has three main components. It has SDS, it has glycerol, it has bromophenol blue. So what is the function of SDS? Just now we discussed the function of SDS, that it is linearizing the protein and it is giving a net negative charge to all your protein, irrespective of the inherent charge which is present on the protein. And the glycerol, what is the function of glycerol? So glycerol adds weight to your protein. So when you are loading the sample into the well, then the sample can leak out of the well. So this glycerol, because it is very viscous, it gives it a particular density. It gives a particular density so that the sample doesn't float away when it is loaded in the well. And bromophenol blue is actually the tracking dye. So yeah, why do you need a tracking dye? So everything is transparent, isn't it? If you do not add a dye, then how will you understand that how much your gel has run? Okay, so that is why you need to add this dye in your sample. So when you run the gel, this by tracking by tracking that what is the position of your dye, you can estimate that how long you have to run the gel. So now let's understand the components which are used for preparing the gel. So first of all is timate. So basically you have to first understand that when you are preparing the gel, so the gel that you prepare, it is actually a catalytic chemical reaction. A catalytic chemical reaction takes place. Okay. So the polyacrylamide gel is formed by catalytic chemical reaction. This is different from an agarose gel that you prepare. So agarose gel is gets solidified only on the basis of temperature. So once you heat the gel, so you dissolve your agarose dissolve your agarose powder in 1x TAE buffer, isn't it? And then you allow it to cool down. So once it gets once this gel is cooled, then the gel gets solidified. But your polyacrylamide gel is acting on a different principle. So a chemical reaction is taking place for formation of this polyacrylamide gel. Okay, so what is this chemical reaction? So the chemical reaction actually takes place between this polyacrylamide and bisacrylamide. So it is a free radical chemical reaction that takes place and bisacrylamide. So for a chemical reaction, what are the different components that will be required? So if you just heat a polyacrylamide you say you have your polyacrylamide solution and you heat it and then you cool it down, then the gel will not be formed. You need to add different components in the yeah, in this reaction. So when you have to actually add different components in this reaction so that this catalytic reaction takes place and the gel is formed. Okay, so for this case, so it is a free radical. So remember that it is a free radical polymerization reaction that takes place. So now who provides the free radical? So the free radical is actually provided by ammonium persulfate. Okay, so ammonium persulfate provides the free radical, and then the acrylamide monomer that is present, that through the free radical reaction, it one monomer joins with another monomer, and then this bisacrylamide will also cross-link these different polyacrylamide monomers, and then your gel will form. Now the catalytic. So for a chemical reaction, you also need a catalyst. So who is the catalyst? So that is TIMET, N tetramethyldiamine. So this is the catalyst that will fasten your reaction. So that will make your reaction faster. And what is the trace cell? Trace function of trace cell is it is providing a particular pH to the gel. Okay, glycine is the component of the running buffer. Bromophenol blue is the tracking dye. Commercy brilliant blue R250 is the staining dye. Is the staining dye. Sodium dodecyl sulfate is denaturing your protein and it is giving negative charge to the protein. And acrylamide are the monomer units to prepare the gel. Bisacrylamide is the cross-linking agent. So remember the function of each of these components. So there are four different components. One is the first is a polyacrylamide. So polyacrylamide is actually a mixture of the acrylamide and bisacrylamide. Okay, so 29 is to 1. In that ratio, it is added weight by weight. So your polyacrylamide is actually the giving will form the base of the gel. So that is what will give the property the gel property. The second thing that you add is the buffer. So your buffer is your trace buffer. So for the dissolving gel you add the trace buffer which is having a pH 8.8. For the stacking gel you add a trace buffer which is having a pH 6.6. Okay, then next component, what is added? SDS. So SDS is added. So SDS will, what will it do? It will linearize the protein and it will give a net negative charge or uniform negative charge to your protein sample. The fourth component that is added is APS. So ammonium persulfate will provide the free radical and that will initiate it will provide the free radical, and when you add TIMET, so TIMET is the catalyst, it will start the free radical reaction. So how the reaction takes place, we will learn in the next slide. Okay. So remember the components. TIMET is your catalyst. APS is your provides the free radical. The trace is providing a pH to the pH to the media, and the SDS is linearizing the protein and giving a net negative charge or uniform negative charge. Okay, so this is your acrylamide. Okay, so now look at this reaction that takes place. So this is your acrylamide and this is the bisacrylamide. So bisacrylamide is basically what it is, N-methylene bisacrylamide. So if you so when you add, so in presence of TIMET and ammonium persulfate, so your ammonium persulfate, actually this bond breaks. This O bond that you see, that is broken and a free radical is produced. Now this free radical will start. So this chemical reaction takes place. This free radical will come and form a free radical here, and then these acrylamide monomers will start polymerizing. Okay. So this is actually your bisacrylamide. You see here. So this is your cross-linking agent. So this is your one sheet of polyacrylamide, second sheet, third sheet, and in between these sheets of polyacrylamide, the bisacrylamide is cross-linking these different sheets of polyacrylamide. Okay, so this is bisacrylamide. So it is a cross-linking agent. So this is how the polyacrylamide matrix looks like. So you see here the pore is produced, isn't it? So you see a pore is produced here, depending on the what concentration of polyacrylamide you are using. So ammonium persulfate in the presence of TIMET forms oxygen-free radicals. Okay, so in the presence of TMAD, it will form oxygen-free radicals and induces the polymerization of acrylamide monomer to form a linear polymer. So it will induce the polymerization reaction and the linear polymer will be produced. Okay. Now in this linear polymer, they are interconnected by cross-linking with bisacrylamide. So these are your bisacrylamide agents and they will cross-link the linear polymer. Okay. And as a result, you get a 3D mesh with pores. Now the size of the pores, remember this, that the size of the pore will be determined by the concentration of the acrylamide. Okay, and the amount of bisacrylamide in the gel. So in a vertical gel electrophoresis, so why is it a vertical gel electrophoresis? So here the flow of electricity is in the vertical direction, so along the direction of gravity. Okay, so that is why it is a vertical gel electrophoresis system. So we cast two types of gel, a stacking gel and a resolving gel. The upper part is called the resolving is called the stacking gel, and the lower part is called the. So this is your stacking gel. And this lower part is sometimes called the resolving gel or the separating gel. Okay. So a thin layer of organic solvent is added. So what happens is when you are preparing your gel, first you add the resolving gel. First you prepare the resolving gel and then you pour it in between the two plates. So you have a thick plate and a thin plate. So between the thick, so you were demonstrated this in the video lecture. So you must have seen that this is poured in between the thick plate and the thin plate. And then once you cast the resolving gel, you pour the resolving gel inside this within these two plates. You add a organic solvent, you add IPA, isopropanol. So why is this isopropanol added? So remember this isopropanol, what it does is because this is a free radical reaction that is taking place, the atmosphere also has oxygen. So to prevent the oxygen present in the atmosphere from hindering this free radical reaction, an IPA layer of IPA is added, and then the gel is allowed to solidify. Okay, so oxygen basically neutralizes the free radical and slow down the polymerization. So these free radicals that are produced by the APS and that is starting the free radical reaction that gets neutralized by the atmospheric oxygen. That is why a layer of IPA is added on top of the resolving gel, and then the gel is allowed to solidify. Okay. So after polymerization of the resolving gel, a stacking gel is poured. So after the gel has solidified, this resolving gel has solidified, we what we do, we discard this IPA. So we uh, remove the IPA, and then after that, what we do, we add the stacking gel. We add the stacking gel and then we place the comb. Okay. So this is how the setup of a SDS-PAGE apparatus look like. So you have a gel cassette, and then you have your electrode chamber. So actually these things are very difficult to make you understand. So once you have go through, you can go through any. So this was demonstrated. So there was a demo video where one of the students demonstrated to you how you can set up the gel apparatus. So you have these plates and then you cast the gel and then you set the gel between this electrode chamber, and then you add the running buffer. So you add the SDS running buffer, and then you remove the comb, and then you pour your samples in these wells. And then you attach this, you connect it to the power supply, and then your sample gets moved through the gel by the process of electrophoresis. Okay. So remember this. So why do you add an organic solvent over the resolving gel? So this is to stop the entry of oxygen. Why do you want to stop the entry of oxygen? Because oxygen, what it does, it neutralizes the free radical and slows down the polymerization. And also another reason to add is it makes the gel crisp. The resolving gel so when you are adding the resolving gel, the surface can have some curves, or it may not be straight. So to make this layer straight, you add the IP as well. So make the top layer smooth actually. So after polymerization of the resolving gel, a stacking gel is poured and the comb is fitted into the gel for construction of different lanes. Okay. If you have the opportunity, then you can visit any lab and you can see how you can set up this SDS-PAGE and how samples are loaded in the well and the gel is run. So now we will understand that what is the purpose of having this buffer of pH 6.8 in the separate in the stacking gel. So the stacking gel has the pH of the trace 6.8 and the separating gel we add the trace buffer which is having a pH 8.8. Okay, so the reason is very simple. So that is something that for understanding this, you need to know the PI of glycine. So what is the PI of glycine? Anyone? Isoelectric point of glycine. So the isoelectric point of glycine is around 6. Okay. So what happens? So your glycine. So basically when you prepare the running buffer, your running buffer also has three components. It has uh, it has 10% SDS, it has tris, and it has glycine. Okay. It has glycine. So now what is the glycine plays a very important role actually. Okay, so what happens? So in the stacking gel has a pH 6.8. Now at pH 6.8, your glycine will be what? Your glycine molecules will have a very less negative charge. It will have a very less negative charge. So most at pH 6.8, your glycine has a very less negative charge. Okay, while your SDS has a very high negative charge. Or your chloride ion. Basically, this is your tris chloride. Okay, tris chloride that you add, your chloride has high negative charge. So what happens? And your protein. So you have three things to understand. So first you have your protein sample. Okay, then you have the glycine molecules. So the glycine molecules are very so the glycine molecules are having a very less negative charge. Okay. And then you have your chloride ions. So chloride ion is negatively charged, highly negatively charged. So in the separating gel, in the stacking gel, what happens? In the stacking gel, your protein gets sandwiched between the glycine molecules and the chloride molecule. And as a result, what happens? So this property allows the protein, all the protein molecules to come at the same level. Okay. So because your protein is getting sandwiched between the glycine molecule and the chloride molecule in the stacking gel, all the proteins will come at the same place in the stacking gel. Now, after the stacking gel, what happens? After the sample crosses the stacking gel, once it will move into the separating gel. Now in the separating gel has a pH of what? It has a pH 8.8. So now since it has a pH 8.8, now this glycine molecules will be highly negatively charged because here the pH is much greater than the PI of the glycine. So the glycine will have a much negative or will have a higher negative charge. So the glycine molecules will start migrating faster than your protein sample. Okay, will start migrating faster than your protein sample, and as a result, your your protein will get separated. Okay. So now let's read and understand this one by one. Okay. So the sample is prepared in the loading dye containing SDS, beta mercaptoethanol, and glycerol. So what you are doing, you have your protein sample. Okay, you have your protein sample. You mix your protein sample with the loading dye. Okay. So your loading dye has three components. One is glycerol, second is beta mercaptoethanol, and third is SDS. So what is the function of beta mercaptoethanol? So beta mercaptoethanol is a reducing agent. So what will it do? So if there is any disulfide bond, SS bond, that will be broken by the beta mercaptoethanol. Okay, what is the function of glycerol? Is your glycerol is providing density to your protein sample. Okay, and then you have the bromophenol blue, which is allowing you to track the movement of your protein sample. Now, as the samples are filled vertically, there is a distance drift between the molecules at the top versus at the bottom in the plane. The problem is taken care once the sample run through the stacking gel. So when you are loading it on the gel, there will be some molecules which are present on the upper surface, some may be present on the lower surface. So there will be so all the protein molecules will not be present at the same place. When you are loading your sample on the well, then all the samples will not be at the same position. So you want all your protein samples to come at the same position. So that is why you need to have a stacking gel. So when the protein will run through the stacking gel, all your protein samples will come at the same position. And how does the stacking gel help all the protein molecules to come at the same position? So basically by this principle. So the pH of the stacking gel is what? It is 6.8. So at 6.8, your glycine is moving slowly in the front, whereas the stress HCl is moving fast. So that is what I was explaining. So when you are doing a vertical electrophoresis, your chloride molecules will run much faster compared to your glycine molecule in the stacking gel. Okay, as a result, what happens? Your sample, your protein sample will get sandwiched. It will get sandwiched between the glycine molecule and the chloride molecule. As a result, get sandwiched, and as a result, what will happen? It will get stacked in the form of a thin band. So in your stacking gel, your sample will get stacked in the form of a thin band. Now, once this sample crosses the stacking gel and moves to the separating gel. So at this in the separating gel, you have pH 8.8. So at pH 8.8, what will happen? Now at pH 8.8, your glycine will have a much higher negative charge. Okay, and so the glycine molecule will start moving faster. And now the sample runs as per their molecular weight. So basically your sample, your glycine, which was present above in the stacking gel. So this was your stacking gel. In the separating gel, what will happen? In the separating gel, you have your sample here and your chloride is over here and your glycine is here. So your glycine moves faster than the chloride and your sample now gets separated according to your molecular weight. Now, after the tracking dye reaches to the bottom of the gel, so how will we know that when to stop the electrophoretic power supply? When to stop the power supply unit? So that is done by tracking the movement of the bromophenol blue. So when you track the movement of the bromophenol blue, you will know that once this tracking dye has left the gel, or it has left the gel, or it has moved to the bottom of the gel, then you stop the power supply and you take out the gel from the glass plate. And then what is done? And then you stain the gel and destain the gel. Okay. So these are the different components of an electrophoretic apparatus. So this is a power supply. These are the wires. These are the wires for connecting. So you will attach the anode to the anode electrode and a cathode wire to the cathode electrode. And then you have an electrophoretic tank. So you place this gel cassette within this electrophoretic tank, and then you add the buffer, the running buffer, and then you switch on the power supply. Okay. So what is the role of timid in gel electrophoresis? Quickly answer these questions. So the TIMET was acting as a catalyst, isn't it? TIMET was acting as a catalyst. So the answer is it catalyzes the acrylamide polymerization reaction. So it catalyzes the free radical reaction or the free radical that is provided by the APS. Okay, so TMAD plays a crucial role in gel electrophoresis. It catalyzes the polymerization of acrylamide. Remember this, it catalyzes the polymerization of acrylamide, and this when mixed with ammonium persulfate, TMAD, what will it do? It will accelerate the conversion of acrylamide and bisacrylamide into polymerized gel through a free radical mechanism. So this rapid polymerization is essential for creating a stable gel that serves as a sieving media, allowing for effective separation. So TMAT acts as the catalyst. Now, what is the function of the stacking gel? Why is it having a pH of what? So what is the pH of the stacking gel? It is asking in this question. So what is the pH of the stacking gel? So remember the pH of the stacking gel is what? It is having a pH of 6.8, and the separating gel has a pH of 8.8. 8.8. Okay. 6.8. So why is it so? So just now we explained, so you can go through this explanation as well. So it is having a pH 6.8 so that it gets sandwiched between the glycine molecule and the chloride molecule, and we can and all the protein molecules come at the same position. Okay. Before moving into the separating gel. So what will the sizes of the pore in SDS-PAGE will be determined by what? It will be determined by the concentration of polyacrylamide. So higher the concentration, the smaller the pore size. Concentration of polyacrylamide. Okay. Concentration will be inversely proportional to the pore size. Okay. The higher the concentration, the lesser the pore size. The lesser the concentration, the higher the pore size or larger the pore size. You can read the explanation. Same thing is written here. The reagent that is used as tracking dye to monitor the progress of gel electrophoresis. What is that reagent that is added? So the reagent is bromophenol blue. Remember, bromophenol blue. So remember the function of each of the components. It is very necessary to remember. Okay, because this SDS-PAGE is done in all labs. In any lab you go, you have to run an SDS-PAGE. Okay, so you should understand the function of each of the component. What is its role? Okay, so the loading. Remember that the loading die, SDS loading die. It is we usually prepare a 5x SDS loading die in the laboratory, and when we add it to the sample, we make it 1x. Okay, so what are the components that are present in loading die? So first is your SDS. Second is what? Second is your beta mercaptoethanol. Beta mercaptoethanol is the reducing agent. It will reduce the disulfide bonds. It will make the protein linear. SDS will also make the protein linear. Next component what is added is glycerol. And third, what you add? You add the tracking dye. Your tracking dye is what? Tracking dye is your bromophenol blue. That will help you to track the mobility or track how much your gel has run. Okay. You do not add the tracking dye, then everything will appear white, transparent. You will not know how much your protein has run. Okay. So you can read this. The same thing has been explained. The sample as it passes through the stacking gel in vertical gel electrophoresis gets what will happen when the sample will pass through the stacking gel. It will get sandwiched between the glycine tris and stacked as a thin band. Okay. So answer is option A. It will get sandwiched between the glycine and the chloride molecules or the tris chloride molecule. So you can read here as well. So it is existing as a Zwitter ion, fast moving tris. So the stacking gel is typically set at lower pH than the separating gel, which causes the glycine to exist as a Zwitter ionic form that slows its migration. So Zwitter ion means what? It will have both a positive charge and the negative charge. So your amino acids are basically Zwitter ions because it has both positive charge, negative charge. So Zwitter ion means what? It has equal number of positive and negative charge that will make it zero. So that is what will happen at the pi of the protein. Now, the differential migration creates an electric field where the sample molecules initially dispersed becomes concentrated into a thin band. In your stacking gel, when the sample will pass through the stacking gel, it will get concentrated in a thin band. Why? Because it is getting sandwiched between this chloride and uh, glycine molecule. Okay. So what is the function of beta mercaptoethanol? So remember beta mercaptoethanol and DTT. These are all reducing agents. What are they? They are reducing agents. So what is the function of a reducing agent? So so in a protein you also have covalent bonds. So the covalent bonds are formed by the disulfide bridges. You know all these things, right? In biochemistry, you must have learned. So the disulfide bridges are formed by what? By which amino acid residue? By cysteine amino acid residue. So these disulfide bonds. So if there is a, so you have this protein sample. So this is your coil helix, and these are your protein. These are your cysteine molecules. So your cysteine is forming a disulfide bond. So when you add beta mercaptoethanol, what will it do? It will convert this SS bond into SH bond. It will get converted to SH, and your disulfide bond will break. So your beta mercaptoethanol and DDT, these two are what? These two are reducing agents. They will reduce the disulfide bond. Okay, they are reducing agents. They will break the covalent bonds. They will break the disulfide bonds. They will reduce the disulfide bonds. So anything you can say. So it will break the disulfide linkages. So what is the staining procedure? So once you take out the gel, then next step, what is the next step? The next step is you add the uh, You take out the gel in a box, and then and then you add the staining solution to it. So this staining solution has, what are the components which are present in a staining solution? Remember, so your staining solution will have a staining dye. So what is the staining dye? It is Kumasi, Kumasi Brilliant Blue R250. This is your Kumasi Brilliant Blue R250. So this will stain your protein sample. Okay. And other than this, what you have glacial acetic acid, and you also have methanol and water. So you need to prepare a staining dye as well. So the methanol, acetic acid, they are all prepared in this composition, 15% volume by volume methanol. So if you add prepare 100 ml staining dye, then you must add 15 ml methanol, 10 ml acetic acid, and the rest you make up with water. Okay, make up with water, and other than this, you will also add add this Comasi Brilliant Blue dye. Okay. So for preparing 100 ml, you usually add 250 grams, milligram, 250 milligram of this Kumasi dye. 10 volumes of Kumasi Brilliant Blue. So you add this, and then you place it on the rocker. So there is a rocker, so that will you know it will shake, have a vertical oscillation, and you keep it for around, so if it is freshly prepared, then 3 to 4 minutes is enough for staining the sample. But if you have used it several times, then you need to keep it for more time. So for 30 to 60 minutes, some people also keep it overnight. But if you prepare fresh staining dye, then then your sample gel gets stained within 3 to 4 minutes. Okay. So there are different types of stain that you can use. So one is this Kumasi Brilliant Blue. This is most commonly used, and it is non-specific. It is one simple staining procedure. Detection limit is 0.3 to 1 microgram. So if your protein amount of protein that you have purified is at least 0.3 to 1 microgram, then this Kumasi can stain the protein. Other than this, you can use colloidal CBB solution, which is 5 to 10 fold more sensitive. Silver stain is 100-fold more sensitive. So even if your protein is very less quantity, very less, you have got very less amount of protein, even then your protein will get stained, because it is 100-fold more sensitive than CBB. So your Comasi can stain 0.3 to 1 microgram. So you divide it by 100. So then around 0.003 microgram. Okay. 0.003 microgram means around 3 nanogram, isn't it? That amount of protein can also be detected. Right. So it is 100-fold more sensitive than CBB. And then you have Cypro Ruby, Cypro Orange, Cypro Tangerine. So sensitivity is less, 4 to 8 nanogram, 2 nanogram, this type of. Okay, these things are clear. Okay, so what is the purpose of Comasi Brilliant Blue? So it is a staining dye. Okay, it will stain your protein. Comasi Blue will stain your protein. How does it stain your protein? So basically, it will only, so after staining, you need to add the destaining solution. So what is done is that you add a staining solution. So your staining solution will contain the Comasi Brilliant Blue. But your destaining solution, after staining for 3 to 5 minutes, you will add the destaining solution. So your destaining solution will also have all the components which are present in staining solution, but it will not have CBB. CBB will not be present. Here CBB will be present. Here CBB will not be present. So when you destain it, then all the non-specific, so the gel will also take up some non-specific stain that will get removed, while the stain which has been taken up by the protein, that will retain. That will be retained. Okay, so the Chromacy Brilliant Blue R250 widely used as staining agent. So the dye binds to protein through ionic and hydrophobic interaction, resulting in a color change that allows for the detection of protein band. So remember this. So it forms ionic and hydrophobic interactions with the protein. As a result, it does not get removed off. So even when you add the destaining solution, your protein band will not be removed, while the extra stain that is there, that will get removed. And it will allow you to detect the protein band. So the reagent that is used to stain glycoprotein in page. So this question we will come later once I discuss the different applications of SDS-PAGE. So how do you do an image analysis from the gel? So basically what is done is when you run a gel, you run a protein ladder along with it. Now this is your dye front. Okay, this is, you will also have a dye front. So dye front is what? Dye front is basically the tracking dye that you add, this bromophenol blue. This bromophenol blue will also will have a dye. You will get a blue colored line at the bottom of the gel. So that is basically your dye front. So why you get a blue colored band, this line, the blue colored dye is line is because of this bromophenol blue. So that is the dye front. That is the how much your dye has moved through the gel. That is the dye front. Now each of these marker proteins will also have a band. Okay. So this one, this band, it is having a molecular weight D1. This band is having molecular weight D2. This is having molecular weight D3, D4, D5, and so on. Now you need to plot a standard. So as I was mentioning, a standard for ACC chromatography, here also you need to plot a standard. So what is the, how will you plot a standard? So on y-axis, you have the relative RF. RF is the relative mobility of the protein with respect to the dye front. And this is the molecular weight. So how do you calculate the RF? So the RF is actually this. You can measure this using a scale as well, or using any analysis software. So how much this has moved compared to this one. So that will give you RF. So RF is basically the migration of the protein band relative to the migration of your dye front. So the distance traveled by the protein. So how much distance this protein has traveled from the top compared to how much the dye has traveled. That will give you the RF. So each of these proteins will have a particular RF value. So your D1 will have a particular RF value, D2 will have another RF value, D3 will have another RF value. So each of these proteins will have have different RF values. So now you can plot this RF. So you know the RF value, you know the molecular weight, you can plot this and you can get a standard curve. Okay. So you can plot it, you can take a graph and you can plot this. So once this is plotted, then this is your sample. Now you want to know the molecular weight of your sample, of the protein band of your sample. So how will you do? Now for this sample also you can get the RF. You can get the RF. So from the standard curve, if you know the RF of your sample, then you can directly use the standard curve and you can get your molecular weight from this graph. Okay. So actually what is done, this is actually log molecular weight is plotted. This log molecular weight is plotted compared to the mean RF values. So this is, for example, this is your RF for your sample. So this has been shown in this figure. So this is the RF of your palette. So you can extrapolate and you can get what is the molecular weight of your protein sample. Okay, is this clear? So migration of the protein from the late divided by migration of the tracking dye. So you plot log molecular mass versus, so you know the molecular mass of each of these protein bands because this is a ladder. So the ladder will, when you use the ladder, all the molecular weights of each of these band will be written. So you can take the log of each of this molecular weight and then you can plot it on the x-axis. Okay. Once you plot it on the x-axis and you get the mean RF, then you can also get the molecular weight of this. So you can get this y equal to mx plus c equation you can plot. Once you plot this curve, you will have a slope, you will have a y-intercept, and you will have the, so your y is your RF value and x is your log molecular weight. So from this equation, if you put the RF value of your protein of interest, from this equation, you can also get the X value. So X will give you the molecular weight of the protein of your interest from this graph. Okay. So is this point here? So this is one of the application of pages that you can determine the molecular weight of your sample. So you can determine the molecular weight by running an SDS page. Okay, by using this RF by calculating the relative mobility. Relative mobility and log molecular weight by plotting the standard curve, you will get to know what is the molecular weight of the sample that you have, protein sample that you have. Okay, now these are some of the points that you need to note down. So when you run a gel, often what happens, it is this actually running a good gel is comes with practice, and also you need to take care of some issues that you face when running a SDS page. So sometimes what happens, you will get some your die front will not come straight. It will give you some curved, smiling like structure. So what are the different cases when such an event can happen? Or sometimes what you do not get very crisp bands. The protein band that you get, that can be diffused. Okay, or instead of getting a single band, you can have multiple bands on the gel, or you can have a streak like your sometimes your protein, you will not get a crease band, but you will have a smear like band on your gel. So what are the different conditions under which you can observe such different gel bands, okay, or protein bands? So why do you get a smiling? So what happens is sometimes uneven heating of the gel. So if you provide a very high voltage, so you are running your gel at, say for example, 150, 170 volt, then what will happen? Your there will be uneven heating of the gel. So the gel which is near to the electrode, those will get heated up. Those will get heated up, and what will happen? How does heat affect the gel? So once you, what will happen if you heat the gel, anything which you heat, your the viscosity of the gel will decrease, so the pore sizes will decrease. So the pore sizes which is towards the end of the gel, they will increase, and as a result the proteins which are present in this in the lanes which are nearer to the left end and the right end, they will migrate much faster compared to the ones which are in the middle of the lane. Okay, middle of the gel. So that is why if there is uneven heating of the gel, your pore size gets increased. Okay, as a result, all the proteins will not move evenly through the gel. Okay, so the outer lanes will move slower than the middle lanes. So rapid heat transfer eliminates this defect and we achieve by filling the lower tank with the buffer until the same height. So either you can place your, sometimes some people what they do, they run the SDS page in ice so that there is even heating. There is no overheating. Okay, or what you can do, you can by filling the lower tank with the buffer until the sample height. So this is your sample height. You add the buffer not only to the base but up to the top. You if you add the buffer, then this eliminates the defect. Okay. The second is what, diffuse protein band. When can you get diffuse protein bands? So if your acrylamide gel that you have prepared, that is not uniform, or if the one portion of the gel has less concentration of acrylamide compared to other portion of the gel, so then in those cases your protein band, we will not get a crisp protein band, you will get a diffuse protein band. Okay. So diffuse protein bands can be corrected by increasing running current by 25 to 50% high, and by using higher concentration of acrylamide. Okay. If you also increase the concentration of the acrylamide, then you will get very sharp bands. Okay. Vertical streaking is what? So if you are overloading the protein sample, then also what will happen? Then you will get smear like bands. Okay. So how will you reduce this? By either reducing amount of protein sample, okay, by reducing a lower amount of protein, or by running the gel at a lower current. Proteins runs faster than expected, so that this actually depends on the property of your protein. If your protein has large number of negative charges or large number of positive charges, then you will never get your protein band at the intended molecular weight. Either it will come much it will come above its molecular weight, so its mobility will be different in the gel. Okay. So it is due to high proportion of basic or acidic amino acids. So this is one thing that even I have seen. So I have one of my protein which is having a large number of basic amino acids, and its molecular weight is around 33 kDa, but it always gives the band around 25 kDa. Okay. So this is dependent on the proportion of basic or acidic amino acids. So that why do you get double bands? So double bands you can get if your protein is getting degraded. if your protein is getting degraded, so for example, you have your C terminus is getting degraded, you can get multiple, you can get double bands, or if there is oxidation. So due to oxidation also you can have double bands. So double bands will be, one band will be your oxidized fraction and another will be your reduced fraction. So that can, how will you resolve that issue? So you add more amount of DDT. You can add DDT and you can more amount of reducing agent so that you do not get double bands. So distorted protein bands, you read these things. Okay, these are something that so how if you get these are some practical problems that we usually see when we are running an SDS page, and you need to understand why this takes place and how you can troubleshoot or how you can resolve this issue. So understanding this thing is very important. So this is a very nice slide where the professor has summarized what are the different issues that we might face and how we can resolve those issues. Okay, lateral spreading, why does this happen? So it can be avoided by reducing the time. So what happens sometimes, what we do, we load the sample and then then we are not running the gel. We are running it after say 15 minutes, 20 minutes. We go and do some other work. So in that case, what will happen? Your protein will get diffused. Your protein will get diffused, and as a result, your protein, you will get a lateral band rather than getting a crisp band here. It will get your band will start diffusing and spreading to the nearby wells. Okay, so that is why once you load the sample, you quickly load the sample and then you switch on the power supply and run the gel. Okay, you should not wait between loading the sample and running the gel. Okay, so appearance of distorted protein bands or uneven protein is due to stacking gel polymerization. It can be corrected by increasing the amount of. So sometimes what happens, you do not get crisp band, you get something like this, or sometimes it is like this. This type of bands also you can observe on the gel. We have also observed it. So if you, so the stacking gel that you prepare, so that the gel concentration, once you pour it, you need to mix it once. If you do not mix it, then then your concentration will not be even, and you will get such type of uneven bands. So if your protein has not come at the same position, then you will not get this. Okay. Now, what is the application for these things? So how can you use polyacrylamide gel electrophoresis? So you can run a urea page as well. So in a urea page, what is done? So from left to right, your urea concentration is increasing. So when you load your protein sample, so you have loaded the same protein sample here, 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 here on all these 10 wells. So these are your 10 wells. But as you are moving from the left lane to the right lane, your urea concentration is increasing. So when you run your protein through this urea gel, what will happen? As the at the higher or the toward the right side lanes will have higher concentration of urea, and your proteins will get unfolded. So you can study protein unfolding by running a urea page. Okay, protein unfolding. So your native protein will be here. So at 0, 1, 1.5, 3, 4, your protein will, but as gradually the urea concentration will increase, you can get uh, the intermediate conformation. So during protein folding, your protein will transition through different conformation. So the native one will be fully folded, but gradually it will unfold. So what are the intermediate states? That also you can actually analyze by running a urea page. Okay. So there is a horizontal gradient of urea, 0 to 8 molar urea. Horizontal gradient of urea is prepared and you run this gel. So what happens? Why is your unfolded protein coming at higher is not able to migrate greater distances? Or why are you observing a lower migration of the unfolded protein? This is because what will happen when your protein is folded? So it is spherical shape. So it will experience less frictional force. But when your protein gets extended, so if your protein has extended now, so it will experience more frictional force. So it will migrate less. Its migration will decrease. Okay. So that is why your unfolded protein is giving bands which are at a higher position compared to your native protein. Because your native protein is folded, it will migrate more distance. So your unfolded protein, it is extended shape. So it will, as it will get unfolded, it will have lesser migration. Okay. Now you can also understand protein-protein interaction. How will you understand protein? You have a protein A which interacts with protein. So this you can only do by running a native page. So in native page, what you do not add SDS. So you cannot add SDS, you cannot add beta mercaptithanol, you cannot add DTT. So all these reducing agents and SDS you have to remove. So when you are running a native page, you are trying to understand the protein in its native conformation. Okay, so once you add SDS, what it is doing, it is linearizing the protein. But if you do not add SDS, then your protein will remain in the native state. It will not get linearized, and and so if you want to study if A protein interacts with B protein, how will you do? So A protein will only interact with B protein in its native stage. If you linearize them, then they will not interact. Okay. So that is why you need to run a native page. So you run a protein A, you run protein B, and then you set up another reaction where you incubate protein A with protein B. And then you run that sample. So if your protein A and protein B forms a complex, then you can see that there is a rise that the complex that is formed that will have a molecular weight which is higher than protein A and higher than protein B. That is why you will get a band which is much higher in position compared to the protein A band and the protein B band. Okay. So you can also do a, this is actually Western blotting only. So you want to identify. So for example, you want to identify that what that so you want to understand that if your cell is expressing a particular protein. So how will you know? So you have treated with some drug, or you have under certain some stress condition you have provided, and you want to understand that under this concept, stress condition, whether my protein is getting overexpressed or whether my protein expression is going down, or under this condition only my protein is getting expressed. Other condition is not. So these type of experiments, if you want to do, you can do a Western blot. So what you do, you lyse the cell, you have the lysate, and then you run the lysate on the gel, and then you probe the probe. So you have some literature that you know that yes, this protein might get. You have a hypothesis that yes, this protein might get expressed under these conditions. So you probe the protein by adding an antibody. So you add an antibody which is specific to that protein. Okay. So that this is actually Western blotting only. You first run the gel, and then you transfer the protein band from the gel onto a nitrocellulose membrane or a PVDF membrane. So once you transfer it to the PVDF membrane, then you add the antibody against the protein, or you can add the protein B as well. So if you want to study protein-protein interaction, you add the protein B, and then you add antibody against the protein B. And then you add the secondary antibody against the primary antibody. This secondary antibody will have a fluorophore, and then you add the substrate, and then you quantify. You prepare the plot, and then you quantify. So if this protein B is interacting with protein A, then you will have the band of protein B. So we will understand this in Western blotting. So another is application of this. So if you want to detect a glycoprotein and phosphoprotein. So for glycoprotein, you add this periodic acid shift reagent. To detect glycoprotein and phosphoprotein, this periodic acid shift reagent specifically stain the glycoprotein. So glycoprotein is what? It means your protein which is attached to some carbohydrate molecule. Some carbohydrate molecule. It can be mannose, it can be some sugar molecule to which is it is attached. So that is a glycoprotein. So how do you detect a glycoprotein? Because your Kumasi will not stain a glycoprotein. It will only stain a protein. So for detecting a glycoprotein, you need to stain it with this reagent, periodic acid shift reagent. And whereas phosphorylated protein can be detected by autoradiography. So P332 is radioactive, so you can detect it by autoradiography. So this is the question that was asked over here. So the reagent, which reagent is used to stain glycoprotein? This periodic acid shift. Okay. Okay, these things I covered. We can quickly go through this. Why does the smiling occur? So due to uneven heating of the gel, the occurrence of diffuse protein band is what? Due to diffuse protein is due to low acrylamide concentration. So if you have low acrylamide concentration, then you will not get a very sharp, it will get diffused. Okay, so low acrylamide, you can read the explanation. Why do you get double band? Either it is getting oxidized or your protein is degrading. So if your protein is degrading, then you will have multiple bands that you can observe below your protein band. Okay. So answer is yeah, this over partial oxidization or degradation. Okay, lateral spreading. When can it happen? So if you have loaded the sample and you have not switched on the power supply, if you are after 15 minutes you are turning on the power supply and running the gel, then your protein will get diffused and it will spread to the nearby bands, nearby wells, planes. Okay, so that is why delayed running option is D. Okay. The primary function of urea is what? Urea is what it is doing in urea page. It is unfolding the protein and it is also dissolving the insoluble protein. So you add urea to any insoluble protein, your urea will dissolve it. Okay. Because urea is a chaotropic agent, it will disrupt the hydrogen bonds, non-covalent interactions, and it will cause them to unfold and adopt a linear conformation. It will linearize it, denaturing agent, and it will also solubilize and unfold and unfold the protein. So the reagent used to strain glycoprotein is what? It is periodic acid Schiff. So the periodic acid Schiff, why does it specifically stain glycoprotein? Because there is some chemical reaction that takes place, carbohydrate moiety. This periodic acid is HiO4, I think. So periodic acid oxidizes the glycosyl residues generating aldehyde groups. The aldehyde reacts with Schiff reagent and a vibrant magenta or pink color is observed. Okay. Remember this. Now, application is of page is that you can do a Western blot. So, so if you, that is what I was explaining. So why do you do a Western blot? So if you want to understand the expression of a protein, or you want to understand, so you have, for example, you have three different cells. You have a liver cell, you have a kidney cell, you have a ovarian cell, and you want to, you want to study the expression of a protein in these three different cells. So you can go for Western blotting, or you want to have treated a particular cell, a cancer cell with different concentration of the drug, and you want to understand with that what is happening to the expression of the protein at increasing concentration of the drug. Then also you can do a Western blotting. Okay, and for you have applied different environments to three different cells. You have given some stress condition, you want to understand that which protein is getting expressed, whether this particular protein is getting expressed in stress condition, then also you can do a Western blotting. Okay, so in Western blotting, what is done? You first run an SDS page, and after running the SDS page, you transfer this protein band from the gel onto the membrane. So this is the PVDF membrane, and then after this, you so the after your protein gets transferred on the membrane, then you add the primary antibody against your protein of interest. Okay. And then after the primary antibody, you wash it off and then you add the secondary antibody. So your secondary antibody will carry the fluorophore, and you can use a substrate against this fluorophore, or you can add some reagent that will react with it, and then you can observe the color. Okay. So only that protein against which the protein against which you have used the antibody, only that band will be visible. The other bands will not be visible. Okay. So it is an analytical technique. If you want to identify the presence of a specific protein in separated sample, then you can do Western blotting. Okay. Or following separation by SDS-PAGE, then what is done? A Western transfer is used to move protein from the polyacrylamide gel. Your protein will move to the membrane. Okay, and then you add the then you incubate it with the primary antibody. So the membranes are probed with antibodies in a process called immunoblotting. So immunoblotting will use antibody protein antibody binding through specific recognition sites. So there are three principal stages. So there are actually four steps. First, you do a transfer, then you do a blotting. Uh, so first you basically run an SDS page. Okay. First you run SDS page, you have to run first. Then you will transfer it on the membrane. Then you will incubate it with the primary antibody. Then you will incubate it with secondary antibody, and then you will develop the blot development. Okay, so these are the four steps. So what are the different materials that you will require? You will require transfer buffer. You will require transfer membrane. You will require a plastic spatula. You need to have the E. coli, the protein sample extracted from the E. coli. Then you will need blotting sheet, semi-dry electro blotting unit. So this is you have different types of electroblotting unit. One is semi-dry. Another you have this liquid also where you transfer it in buffer actually. Reagents for performing semi-dry electroblotting unit, then reagents for performing electrophoresis and anti-GFP antibody, anti-rabbit IgG HRP. So this is your secondary antibody. This is your primary antibody. And then there are the developing reagents. So you need to have the substrate and these different chemicals which can react with the fluorophore or the substrate or the enzyme which is present on the secondary antibody. So what is done? So the first step will be what you have to prepare your sample. So if so for solid tissue, what you will do, you will homogenize it, such as it can be a tumor or a whole liver, it can be brain. So it is more mechanically broken down by passing it through a blender or homogenizer or sonicator, and then the lysate that you get. Once you have the lysate, or if it is an individual cell, then what will you do? You will directly incubate it with lysis buffer. So your lysis buffer has different detergents, REPA buffer and all these different buffer you have that will actually lyse the cell membrane, and all the proteins which are present in the inside the cell, they will come out. Okay. And also you need to add this phosphatase and protease inhibitor so that they are these protease and phosphatase which are present in your cell, they do not degrade your protein of interest. So in the first step, the protein is transferred. So first you need to prepare your samples. Step one. Step two is what you need to do an electrophoresis. So why will you do an electrophoresis? So what will your electrophoresis do? It will separate. So your lysate has different proteins. Okay. So it will separate all these different proteins based on their molecular weight. Okay. Now once you have separated the different proteins, then you will do a transfer. So in this transfer, what will happen? When this transfer is done by your the protein which is present on your gel, that will get transferred on your membrane. That will get transferred for you. So for this, you need to set up the transfer unit. So you have to cut the membrane. You have to that is of the same size as the gel. You have to cut it in the same size. Then you need a nitrocellulose membrane. So the place the membrane in the transport buffer and observe that liquid has with the membrane areas upright as well. So then you need to, for PVDF membrane, you need to use 100% methanol. So remember this. So for activating the membrane, you need to immerse it for 5 minutes or 7 minutes in 100% methanol, 15 to 30 minutes, and then decant the methanol. For PVDF membrane, you need to activate it by immersing it in methanol. But for other membranes, you can your nitrocellulose membrane, you can directly place it on the transfer buffer, and then you need to assemble the transfer cassette. So how will you assemble the transfer cassette? So see this figure. So this is your anode electrode plate. So this is how a semi-dry electroblotting unit looks like. Okay, so this was mentioned, isn't it? Semi-dry electroblotting unit. So this is how it looks like. So you have this anode electrode, and here you place some filter paper. So filter papers are basically this white colored blotting sheets actually. So you place this filter paper, then on top of the filter paper, you need to place your nitrocellulose membrane. Above the nitrocellulose membrane, you place your gel. Above the gel, you place again a few sheets of filter paper, and then you place the cathode. So what will happen when there will be flow of electricity, then the protein sample which is present on your gel, it will get transferred to the nitrocellulose membrane once you apply the power supply. Okay, so here the steps have been written one by one. So you remove the stacking gel, you equilibrate the gel in transfer buffer, then place a pair of blotting sheet on the anode plate. So this is your anode plate. You have to place the blotting sheets on top of it. So this is how you have to assemble the transfer cassette. Then after that, you place the transfer membrane on the top of the blotting sheet. So once you place the blotting sheet, then you place the PVDF membrane, then you place the gel, then again you place the blotting sheet. Okay, so you have four things. So you have the anode plate, then you place the blotting sheet, then you place the gel, then you no, so then you place the PVDF membrane, then you place the gel, and then you again place the blotting sheet and place the cathode. Okay, and after that you switch on the power supply. So you will run it at 100 volts for 1 hour, and then your protein will get transferred from gel to the membrane. So then after that, once it has been transferred on the membrane, then the next step what you do, in the next step you have to add this one. This is very important. So first you stain it with Ponsu. So why do you stain it with Ponzu? So to understand whether the transfer has even taken place, whether your protein bands have been transferred from your gel to your PVDF membrane. So you need to stain it with Ponzu. That will give you the different bands. You can see the different bands of protein. Now after that, after you stain it with Ponzu, you wash it off several times and then you add this blocking buffer. So you add this 5% skimmed milk, or you can add 5% fat-free BSA. So why do you need to block it? So you do not want your primary antibody to attach anywhere on the membrane. So that is why you need to you add this 5% skim milk or 5% BSA, so that it will create a layer on that PVDF membrane, and only where your protein is there, only at that region your antibody will go and bind. So in the blocking agent is added, and then in that, so there are two-step probing. You can add two-step probing, or you can do one-step probing. So in two-step probing, what is that? First, a primary antibody is against the protein you want to probe, and then a secondary antibody is added. Okay, so two-step probing means you add a primary antibody, then you wash it off after incubating it for overnight, and then you add the secondary antibody. But in one-step probing, what is done? Only the primary antibody, which is conjugated with an enzyme, that is added. But this one-step probing is not done. This two-step probing is only done because one-step probing is can give you non-specific bands. Okay, so your one-step probing is usually not done because uh, this is the resolution is not very good with this, or you can get non-specific band. This is more specificity increases. Now for blot development, depending on what type of secondary antibody you are using, whether your secondary antibody is conjugated to horseradish peroxidase, whether it is conjugated to alkaline phosphatase, whether it is luminescent, whether it is HRP or alkaline phosphatase base phosphatase, depending on depending on what is your secondary antibody, or which fluorophore is tagged to your secondary antibody, accordingly you have to add your reagent for blot development. So if it is HRP base, HRP is what? It is an enzyme which is horseradish peroxidase. So for that, what reagent is added? 4-chloronaphthol or DAB or TMB. So if you add fluoronaphthol, then you will, what color you will get? Purple precipitate. If you add DAB, then you will get brown colored precipitate. If you add TMB, you will get dark purple stain. If it is an alkaline phosphatase conjugated, then you need to add BCI, this BCIP. And what color you will get? Indigo precipitate. So you can read about this. These are information, some points that you need to remember. Okay. So colorimetric detection, or you can do a chemiluminescent detection. So colorimetric detection is wash the membrane with TBS to remove detergent, place the membrane into the colorimetric reagent, and protein up bands will appear in 10 to 13 minutes. Then or if it is chemiluminescent, you can transfer the membrane into this chemiluminescent reagent, then soak the membrane for 30 seconds to 5 minutes, drain off the reagent and wrap the membrane into the plastic wrap. Or for fluorescent detection, you do the secondary antibodies. Okay, so these are the various steps. First, it is separated by electrophoresis. The protein bands are separated. Then from the gel, it is transferred onto the sheet, from to the PVDF sheet, and then you add the primary antibody. Then after that, you do washing steps to remove the non-specific binding, and then you add the secondary antibody, and again you do the washing, and then you add the photographic film. You do imaging, or take the imaging blot development. You do autoradiography. So which reagent is used to block membrane in Western blotting? So which is the reagent that is used? It is either skimmed milk or free fatty acid BSA. Free fatty acid BSA is added. Okay. So skimmed milk, why is it is added? To prevent the non-specific binding of the antibody to the membrane. Skim milk is commonly used as blocking reagent. It will prevent non-specific binding of antibody to the membrane after transferring protein to the membrane. It is crucial to block any unoccupied site to reduce background noise during detection. So how does skim milk help in that process? Because skim milk contains casein. Casein is a protein which binds to the membrane surface and occupies these potential binding sites without interfering with specific interaction between the target protein and primary antibody. So your skip milk contains casein, and casein will interact with your membrane and block the potential binding site. And as a result, but it will not interfere with the interaction between the protein and the antibody. So that is all for today. So is there any doubt that you couldn't understand? Anything that you want more explanation? So, so that was all for today. So if there are no more doubts, then we can stop today's session and then we can again meet in the next week. Thank you all.